Please find a seat. We'll get started soon. So welcome to our panel discussion on neural organoids and chimeras. If you're anything like me, a few years ago, I had no idea what these things were. And many scientists and philosophers have not spent too much time thinking about them yet. So hopefully we can have a fun discussion engaging some cutting edge bioethical issues here. My name's Dylan Stull. I'm a medical student at Stanford University in my final year. I've spent a couple years studying these things, but it's an absolute honor to have the privilege of introducing this distinguished panel, although one of our members has admitted that he agreed to be on the panel quite begrudgingly. I won't say who. In order to avoid interrupting the flow of our ideas once we get going, I'm going to introduce everybody right now at the beginning. I'll start off with Kristen Collier, who's going to be kicking us off with some remarks from the University of Michigan. She just got promoted to Associate Professor of Internal Medicine at the University of Michigan in Ann Arbor, where she serves as the director of the University of Michigan Med School Program on Health, Spirituality, and Religion. Her special clinical interests include preventative medicine, primary care, depression, and heart disease. Her scholarly work explores the overlap of spirituality, religion, and medicine. Her work has appeared in JAMA Internal Medicine, the Annals of Internal Medicine, the Journal of General Internal Medicine, and the American Journal of Hospice and Palliative Medicine. Dr. Collier's work has also appeared in Notre Dame's Church Life Journal, Theopolis, and America Magazine. Welcome, Dr. Collier, to the panel. Second, I want to introduce Dr. Charlie Camosi, Professor of Medical Humanities at the Creighton University School of Medicine. Also, congratulations on this very recent appointment. He's the author of numerous scholarly and popular articles, as well as 4 books. Too Expensive to Treat was a 2011 award winner with the Catholic Media Association. Peter Singer in Christian Ethics was named a 2012 Best Book with ABC's Religion and Ethics, and For Love of Animals was featured in the Beliefs section of the New York Times. Most recently, Professor Camosi is the author of Losing Our Dignity: How Secularized Medicine is Undermining Fundamental Human Equality, published by New City Press. He's spending the 2022-2023 academic year as the Monsignor Michael J. Curran Fellow in Moral Theology at St. Joseph Seminary. Welcome to the panel. And I think he needs no introduction, but Carter Sneed is a professor of law at the Notre Dame Law School, concurrent professor of political science at the University of Notre Dame, and the director of Notre Dame's wonderful De Nicola Center for Ethics and Culture. He's one of the world's leading experts on public bioethics, the governance of science, medicine, and biotechnology in the name of ethical goods. His research explores issues relating to neuroethics, enhancement, human embryo research, assisted reproduction, abortion, and end-of-life decision-making. He's most recently the author of What It Means to Be Human: The Case for the Body in Public Bioethics, published by Harvard University Press. Named one of the 10 best books of the year by the Wall Street Journal, and winner of the 2021 Expanded Reason Award. So we'll start off by hearing from Dr. Collier, some remarks. We'll transition to hear from Dr. Camosi and Dr. Sneed, and then we'll have some time for general discussion, Q&A. So please have any questions ready.
Thanks, Dylan. Thanks everyone for joining us. So welcome to our panel discussion on human neural organoids and chimeras. I'm grateful to be on this panel with 2 wonderful friends and colleagues, and together we, along with you, will explore the newer and emerging science of human neural organoids, human neural transplants, and human neural chimeras. We recognize some of you may not be all familiar with this topic, and some of you are content experts. I'm looking at our moderator right here. But in order for everyone to have at least some background knowledge, I will first make the first set of remarks. I've been asked to set the scientific table, so to speak, with the goal of my remarks being to provide some baseline information about the science, explain the rationale behind this type of research, and finally pose some of the questions this emerging science raises that we will then explore with the help of Dr. Kamosi and Professor Sneed, and I hope Dylan as well. When I was preparing these remarks and thinking about this most challenging topic, I wanted to keep both human beings and animals in the forefront of my mind. I found it helpful to remember that in the Genesis account, humans and land animals are described as being created on the same day, and to remember that both are known and loved by God. I would like for us as Christians to consider what it means to honor both animals and human beings alike as we do research to help human beings who are profoundly suffering from neuropsychiatric diseases, while at the same time respecting animals' inherent worth as God's creation whom he loves. And with this in mind, I'd like to dedicate my paper to the humans and the animal that I kept in the forefront of my mind as I worked on this topic. First, to my patients with serious, life-limiting, and life-altering diseases of the brain. And second, to the dedication of the memory of an animal that I know has loved and made a great impact in the life of someone on our panel, that of Charlie's sheepdog Clancy that he had as a child. We are here in recognition of both human beings and animals, and I'm looking forward to discussing how to honor the inherent value of both of these creatures as the very type of creature that they are, made by God, in light of the very urgent need for better understanding and treatments of diseases of the brain. And so first, let me provide some background. Recognizing that there are many non-biomedical scientists in the room, and thank goodness for you, by the way, I want to provide some very basic definitions of what we mean when we even talk about human neural organoids and chimera. So at the most very basic level, these are models by which to study the human brain, either by growing human brain cells in the laboratory in which they are organized in a way that resembles a very simple three-dimensional brain, or putting some human brain cells into another animal, which could then grow and integrate in that animal brain and then be able to be studied. And I will go into much more detail of these different models in a bit, but before I do, I want to explain why we would need such models in the first place. Well, the reason is that these diseases that affect the brain are very common and devastating, and the current ways in which we study the human brain are limited, which has restricted our ability to understand these diseases and have better treatments. So what diseases am I talking about? Well, diseases that we often classify as neurological or psychiatric, diseases like Alzheimer's or other dementias, other neurodegenerative diseases like ALS, demyelinating diseases like MS, Parkinson's, psychiatric diseases such as schizophrenia, and even other conditions that are very common that affect the brain, like autism spectrum disorder. So we know from the numbers that tens of millions of individuals in the US alone each year suffer from neurodegenerative diseases. And when you consider both neurological and psychiatric disorders together, they together represent the leading cause of morbidity worldwide. And each year, neurological and psychiatric diseases in the US are estimated to cost billions of dollars from both costs related to lost productivity, but also as well as clinical care. So because of the morbidity and mortality of these diseases, the NIH has made brain research a priority, budgeting billions of dollars to help our understanding of diseases of the brain. But it's one thing to hear these numbers in the abstract, but another thing altogether to know or love somebody who has a disorder of the brain, or to have a patient look you in the eye who has ALS or progressive dementia who says, please do something. We must act to help reduce suffering, slow disease progression, and improve our treatments for these diseases, but how best to do that? And at what cost? So you might be asking, can't we and don't we already study the brain? What about our usual research on mice and other animals? What about functional MRI or post-mortem examination of the brain? Can't we understand the brain through mechanisms we already have without putting human brain cells into other animals? Well, yes and no. We absolutely have some insight into the brain already. Things like an electroencephalogram or EEG, or functional magnetic resonance imaging like an fMRI, are non-invasive ways to study the brain. However, they cannot give physiological information really, and the types of experimental manipulations of the brain that you can study with them are very limited. The post-mortem examination of the brain has given us and will continue to give us some insights. But to be simple, a dead brain's not a living brain. And so again, the ability to access important information or to do testing in an active way is very limited. Because the location of the brain within the skull and the risks of brain research and the limitations around it, because of important ethical guidelines for live subjects, our ability to fully study the brain is limited in important ways. And because of this, and also in part because the brain is the most complex of all the human organs, advances in understanding how the brain works has really lagged behind compared to our understanding of other organ systems. And investigating the molecular and cellular basis of brain function requires access to living, real brain tissue for reasons above have been limited. Well, what about animals? Well, whatever you think about regular animal research, let alone the chimeras that I'm about to describe, this research has been extremely valuable, but again, it's quite limited. The brains of murine models, namely rats and mice, differ in important ways from human brain cells, and that's often why therapies and discoveries that seem very promising that you hear about that come from research on murine models often really fail to translate when you apply them to humans. And also importantly, murine models are most helpful in modeling diseases that are caused by mutations in single genes, which can be manipulated into the mouse genome. But most common human brain diseases are polygenic, and which means that their cause is from different effects of many variants in genes, and these genotypes cannot be made in murine models. And lastly, many of the disorders we want to study are caused by problems in areas of the brain that are challenging to study in mice. For example, diseases that affect behavior rising from the frontal cortex. The frontal cortex is not as developed in mice as it is in humans. And the behaviors that result from perturbations within the frontal cortex can't really be seen in mice and rats to be observed. And so what to do? Well, this is where the human neural organoids, transplants, and chimeras come into the picture. These, again, are ways to study living human brain tissues in novel ways. And because I'm just a lowly medical doctor who I've been told knows how to assess the body as a set of pipes, tubes, and circuits, I will leverage that skill now to try to explain this science in the simplest way I know how. And then we will end with posing some questions that our ethicists will help us think through. These are the difficult tasks. Okay, so let's start first by briefly talking about these models. Their very basic building blocks are stem cells. And just to be sure everyone's up to speed on how we get stem cells for the types of research I want to review, quickly the various types that are used to clarify any misconceptions that you all have. So for a basic definition, stem cells are undifferentiated cells from an organism which have the potential to give rise to indefinitely more cells of the same type, and from which certain other kinds of cells arise by differentiation. So where do they come from? I think if you ask most lay people, when they think of stem cells, they think of ESCs, or embryonic stem cells. These are obtained from early embryos at a time when the cells are still totipotent, or capable of differentiating into any cell type. So the use of these cells, or at least so, has generated much controversy, especially in the religious community. There are also adult stem cells, which have a more limited ability to give rise to various cells of the body, although emerging evidence is challenging this. But they are found in small numbers in adult tissues, mostly in your marrow and fat. And then in 2006, a remarkable discovery was made by a Japanese researcher, Shinya Yamanaka, when he discovered that through a progress of genetic reprogramming, you can take mature adult cells, so somatic cells, and reprogram them back to become pluripotent again. And they therefore act similarly to ESCs, or embryonic stem cells. And he was, if you missed it, he was awarded the Nobel Prize along with Sir John Gurdon for this work in 2012. So these cells are known as induced pluripotent stem cells, or iPSCs. So these cells are usually obtained through a skin biopsy from an adult, and then they are sort of reprogrammed, and they can be used as an alternative to ESCs. So the models I'm going to try to explain to you briefly all use pluripotent stem cells. And let's start first with human neural organoids. So human neural organoids are three-dimensional aggregates of human brain cells grown in the laboratory from stem cells. The stem cells are given a bath of sorts in the right type of environment to give them what's called a neural induction, to have them develop into brain cells. And then they are guided to develop into a certain part of the brain. These are called brain region-specific organoids, or pre-patterned organoids. Or they cannot be guided into a specific part, but left to develop, so they provide a very rudimentary model of a whole brain that is three-dimensional. These are known as self-patterning or whole brain organoids. And they can provide some insights to the molecular, cellular, and developmental features of the fetal brain. Or you can combine many different brain region-specific organoids in close proximity to form what's called an assembloid. And unlike conventional monolayer cell culture, these organoids can contain multiple neuronal and glial cell types. And unlike classical neuronal cultures, they can exhibit synaptic interactions among the types. So these organoids are very small. They're usually no more than about 4 millimeters or so, and they're very limited in their maturity and complexity, but folks are working to overcome this. So the second model is known as human neural transplants. So this is when human cells are transplanted into the brains of animals during development or adulthood and under conditions that favor their differentiation into neurons or glia. And so you therefore will then be able to study human brain cells in the context of a whole behaving live organism, like a mouse, for example. However, the ability of these human neural cells to mature and integrate within the host brain are limited, mostly in part because human brain cells mature much more slowly than, for example, mouse counterparts. And this results in like a developmental mismatch of sorts. So that then affects the potential for the human neural cells to sort of really integrate within the circuits of the host brain. But in a successful transplant, the human neural cells would integrate into the host nervous system of the animal, receiving synaptic inputs from and providing synaptic inputs to host neurons. And finally, human neural chimeras can be thought of as a special case of human neural transplants. And so for these, the human stem cells would be transplanted into the animal host very early on in developmental embryonic development. And then these cells would intermingle with the host cells as the brain develops, thereby populating it from the very earliest stage and developing in parallel with the host, providing a really high level of integration. And in one particular method called blastocyst complementation, the transplanted human stem cells would replace many of the host cells in a particular region of the brain, so that the part of the brain in the animal would be almost entirely derived from the human stem cells. The blastocyst, just to remind you, is a hollow ball of cells within which a small group called the inner cell mass is destined to give rise to the entire embryo. And also, by the way, the cells that form the ball itself are precursors of the placenta. So with this technique, the stem cells are injected into the blastocyst cavity where they mix with the animal cells of the inner cell mass, and then this can be implanted into the uterus of a female of the host animal species where it can develop and give rise to a live chimeric offspring. To date, chimeras that develop to fetal stages or later have only been generated really mostly using rodent stem cells placed in rodent hosts, but research in this area is under investigation and advancing rapidly. So what I've described to you likely, hopefully, has engendered a lot of important questions. And I'd like to begin the last section quickly of my paper as we give voice to some of the questions and concerns that you likely have and that Carter and Charlie will help us think through with a line from Humanae Vitae. While this line was written in the context of artificial contraception, I think its truth and applicability are worth noting in our current context. Quote, "The church is the first to praise and commend the application of human intelligence to an activity in which a rational creature such as man is so closely associated with his creator. But she affirms that this must be done within the limits of the order of reality established by God," end quote. So our question today is, what are the limits of the order of reality established by God? Will there be objections from those who can see only the promise, maybe an illusion, of these scientific models and not see the risk of theological trespass? As Pope Francis writes in Lumen Fide, quote, "We can almost hear the objections of many of our contemporaries. In modernity, that light might have been considered sufficient for societies of old, but was felt to be of no use for new times. For humanity come of age, proud of its rationality and anxious to explore the future in novel ways," end quote. Because as soon as you start talking about limits to this type of research, one that has the potential to help relieve suffering of millions of human beings, you might be accused of being anti-science. But as Dr. Camosi writes in his wonderful book, For the Love of Animals, quote, "It is not anti-science to discipline medical research with ethical principles and rules. It is actually pro-science to make it consistent with the principles of justice and nonviolence," end quote. Humanae Vitae also states, quote, "An action which of its very nature contradicts the moral order must therefore be judged unworthy of man, despite an attention, quote, to promote the welfare of an individual, a family, or of a society in general," end quote. So even with the needs very clear, do the ends justify the means, especially when we are dealing with human beings and animals, both of whom have moral status and are loved by God? And as St. Aquinas would have us ask, is this choice worthy? Not just what happens as a result when all is said and done. And you may ask, finally, isn't there already oversight into the treatment of animals used in research? Yes, but like any government sort of oversight, it's spotty and uneven, and this type of research is so uniquely different that a different approach must be and should be in order. And so with this in mind, the National Academy of Science, Engineering, and Medicine actually has a committee now on ethical, legal, and regulatory issues associated with neural chimeras and organoids, and had a hearing a couple years ago where they invited experts from a variety of disciplines to provide their perspective on this topic before they drafted the consensus report that's now available to everybody. It was so encouraging to hear that the committee included veterinarians, moral theologians, and ethicists, and voices from the theological community. In fact, one of them is with us today, our own highly esteemed Dr. Kamosi. So the questions that I'll pose to you now and that we'll in large part discuss today are the following. As these organoids become larger and more complex, could they acquire aspects of human consciousness? As researchers successfully integrate more human cells into the brains of animals, could the resulting animal have capacities substantially different from those typical of their species? And if so, would these animals need to be treated or when the time comes discarded in a different manner than other just routine lab animals? And do we have to adjust the way we do consent for use of these cells from humans to be used in this particular type of research? And do chimeras violate a distinction that we hold as sacred between humans and other animals? Or in other words, constitute a theological trespass of sorts? Is this research an affront to both human dignity and an affront to animals who too have value? How do we consider the pressing needs of the public who suffer greatly with neuropsychiatric disorders with the ethical issues this research poses? Importantly as well, I think it's important to give voice here that the public has not been really heard on this subject, and I wonder what the average person would think of this research. Would they think this research demonstrates a kind of hubris or unholy mixing of species, one where the scientist is playing God? The United States currently lacks robust mechanisms for facilitating this much needed type of public engagement, and the public often wants to be informed and to have a say in matters such as these. Social science research on public attitudes toward perspectives on chimeras is currently lacking in the US. This would make a fabulous research project, by the way. And finally, what does the Catechism say about research on animals? It says, quote, medical and scientific experimentation on animals is a morally acceptable practice if it remains within reasonable limits and contributes to caring for or saving human lives, end quote. That's the Catholic Catechism, by the way, sorry I didn't say. But of course, the phrase reasonable limits is open to interpretation and one that we will ponder as a panel. It also goes on to say that, quote, it is contrary to human dignity to cause animals to suffer or die needlessly, end quote. This to me was such an interesting line when I just read it for the first time recently. It's contrary to human dignity to cause animals to suffer or die needlessly. What does that mean? From my reflection on this and from what I've learned over the years from people like Dr. Kamosi is that we are intertwined with the animals in a special way in what Pope Benedict claimed in Caritas and Veritate is a covenant of sorts that flows out of a special concern for everything God has made. God has created all things as interdependent, and we are told this in part in Ecclesiastes 3:18, quote, for the fate of human beings and the fate of animals is the same. As one dies, so dies the other. They all have the same breath, end quote. So let us continue to reflect on God's created order, his creation, human beings and animals alike, as we ponder the questions raised by the science of human neural organoids and chimera in light of the theme of the conference. Thank you.
Good afternoon. I normally like to start with a funny little quip, but it turns out that neural organoids and chimeras aren't as funny as you might think at first.
They're kind of funny.
So maybe Carter will be able to help with that one. I got nothing.
I got one for you.
Okay. I must admit that, especially given this panel's topic and expertise, I have a little bit of imposter syndrome, given the expertise, particularly medical expertise and biological expertise to my right and to my left. And if I'm not mistaken, Carter is writing a whole chapter in a book on this topic. Is that right? That's the idea. So I hope I can offer something of value as a moral theologian here, especially in thinking about the moral status and treatment of non-human animals, which is something I've thought about quite a lot. So the importance of the therapeutic goals behind the kind of research that Dr. Collier so helpfully walked us through just now can hardly be overstated. Indeed, the urgency surrounding them is such that I think it is clear we are not doing nearly enough to achieve them. The coming explosion of neurological diseases constitute a hidden, ticking cultural time bomb that we are in no way ready to have go off. Indeed, at last year's conference, I argued that we need an all-hands-on-deck approach when it comes to dementia in particular. We are already putting 1 in 5 nursing home residents with dementia in a kind of chemical straitjacket through the use of off-label use of antipsychotics, doubling their chance of death, and essentially keeping them in an inhuman state for whatever life they have left. What is going to happen when the number of people with dementia doubles over the next 20 years? What is going to happen when it triples over the next 30 years? In that talk, I argued that we need the church to step up and be the church and totally reorient ourselves to care for this population to avoid the coming disaster, particularly in a dominant secular culture that is likely to deem them non-persons. It would be better, of course, to find cures and therapies for these kinds of neurological diseases. The temptation in this kind of desperate situation, of course, however, is to put aside clear ethical boundaries in order to achieve an important and time-sensitive set of goals. The temptation, put more precisely, is to default to the consequentialist reasoning Dr. Collier mentioned and leave out or minimize questions of intrinsic value, justice, and exceptionless moral norms. Though in some ways, medical ethics was born by attempting to foreground questions of intrinsic value, justice, and exceptionless moral norms, from Nuremberg to Tuskegee and on and on. They resisted straight-up no-chaser consequentialist reasoning very directly. I don't think that the primary ethical issues associated with these practices, however, are risks about consciousness. Even if we were to somehow create full brains, this does not present a risk for them becoming conscious. Fun brain in a vat on Mars, thought experiments notwithstanding. There are thinkers at this conference who are much better trained than I am in the philosophy of mind and related fields, but I think I feel comfortable saying human consciousness, much to the chagrin of those who would prefer to reduce our fundamental reality to the neurological, cannot be found or located in the brain. Indeed, the best theories of human consciousness today, as far as I can tell anyway, think of consciousness as a kind of mysterious product of the human organism itself, holistically considered interacting with its environment. Hylomorphism. No, in my view, the primary ethical issues which are present here come from concern about animal ethics and creation care, which is why I'm so excited to be at a conference with this theme talking about the chimeric research being proposed. It couldn't fit any better in my view. I want to argue that we have a moral duty of justice to animals, which means following God's creative will for particular animals in their particularity. For that animal to be the kind of creature that God created them to be, from housing to companionship to stimulation to exercise and much more. Folks with my view should admit that this is admittedly difficult to do, especially in light of what's just been presented, the great and urgent need to develop therapies for human neurological diseases, and because taking the more contemporary work on animal ethics seriously may mean fewer good research outcomes, at least in the short run. But holding up these stop signs is an absolutely essential part of what medical ethics was born to do and is supposed to do, at least if medical ethics doesn't simply roll over for consequentialism. But doesn't Catholic teaching explicitly say, as Dr. Collier reminded us, that we can use animals in research, right? Paragraph 2417 of the Catechism of the Catholic Church insists again that medical and scientific experimentation on animals is morally acceptable, right? But wait for the conditional. As she helpfully pointed out, if it remains within reasonable limits and contributes to caring for saving human lives. How does the Catechism imagine such reasonable limits? Well, one explicit claim is that because animals belong to God, not us, and are under God's providential care, we human beings, quote, owe them kindness. Owe them kindness. Notice that the language of justice is used here. What do we owe animals? We owe them kindness. And kindness, of course, is not just some anodyne or milquetoast niceness. On the contrary, kindness, both Scripture and tradition tell us, is one of the key fruits of the Holy Spirit. One, frankly, which requires the development of other very important virtues like compassion, courage, forbearance, mercy, and more. If one has this particular gift of the Holy Spirit governing how we treat animals, this does, of course, mean we should be concerned about pain and suffering. But this bare minimum moral concern is where a utilitarian-dominated medical ethics discourse puts its near exclusive focus. Especially in a Catholic context, kindness to animals requires going well beyond this, including respecting the God-given telos of non-human animals in a much broader sense. In St. John Paul II's great social encyclical, Solicitudo Rei Socialis, he insists that, quote, dominion granted to man by the Creator is not an absolute power, nor can one speak of freedom to use and misuse or dispose of things as one pleases, end quote. That's number 34. In Caritas and Veritate, Benedict XVI adds that we must avoid aiming, quote, at total technical domination over nature, because the natural environment is more than just raw material to be manipulated at our pleasure. It is a wondrous work of the Creator containing a grammar which sets forth ends and criteria for wise use, not its reckless exploitation, end quote. That's number 48. Part of what it means to understand the grammar of creation is understanding that each living creature has an intrinsic nature, the pursuit of which results in that creature's flourishing. An animal is different from a hammer, of course, because a hammer has been created for one purpose, to be a tool for human beings. It has no intrinsic goodness or end apart from that instrumental value. By contrast, God created animals good, period, to flourish in their own right, according to their own natural kind he created them to be. Each animal has her own nature, or telos, which God intends her to achieve in her fullness. Indeed, Scripture explains that ultimate salvation will result in a new heaven and a new earth in which all creatures will be redeemed and live out their most flourishing selves, right? We've all heard about lambs laying down with lions and babies with snakes. David Clough, whose work in animal theology and ethics is of the utmost importance in my view, told the following story at a conference presentation I was at Oxford that I helped organize a few years ago. I think it is a quite beautiful way to think about an animal's telos. He said, quote, last week I found a young bird dead at our doorstep. My response was one of sadness, not because the bird at that moment of its death had a preference for its life to continue, nor because its parents would currently be grieving its loss, neither of which may be true, but because I had a sense of the life ahead of this poor creature, of its growth to maturity and the particular contribution it should have made to the universe of creaturely life. The death of that young bird is sad not because preferences went unmet or the sum of happiness was infinitesimally diminished, but because this one life did not reach the end to which it was ordered, end quote. It is one thing to mourn this when it happens in a fallen natural world fundamentally damaged by sin, but it's quite another when we ourselves directly participate in and even create this disorder ourselves. Many of you are no doubt aware of the horror show that exists in many intensive factory farms today. Like chickens bred with bodies so big that they have chronic pain in their legs their entire lives, often to the point where they cannot stand up and they live most of their lives in their own feces. Turkeys, appropriate, I'm sorry for this time of year, bred with breasts so large that they are physically incapable of having sex and require human intervention to procure the male semen and blast it into the female. For those of you that may have been a fan of the show Dirty Jobs, you may remember there was a whole episode just on this one, as you might imagine. Or how about using biotechnology to remove the feeling of satiation of pigs so they eat so much and as quickly as possible, getting as big as quickly as possible, while never even having the modest relief of a full stomach. All done in the name of maximizing protein units per square foot. And what about the context of medical research, which admittedly has a much better end in mind, but we should think about the means as well. Like the creation of billions of mice with specific cancers on which we use to test different cancer-specific drugs. Maybe you've seen some of the biotech websites which have a literal make my mouse box you can push with drop-down menus allowing for knock-in and knock-out various genes that you can just get delivered to your lab, maybe like a kind of Uber Eats. Researchers are now able to modify pig embryos in the womb so that their pancreas never develops, in the hopes that these animals will be useful for developing future organ transplant procedures. Similarly, scientists studying dementia can purchase rat Alzheimer's models from commercial laboratories. Prior to shipment, a slow-release pump system is inserted into the rat's brain, injecting toxic compounds over 4 weeks. The resulting brain damage is said to mimic Alzheimer's symptoms. I propose that even if done for very good ends, these means are not consistent with the kind of stewardship God has entrusted us to exhibit in his caring for creation. Kindness means that our use of animals in medical research cannot be simply about power. It cannot be a context in which we simply treat an animal like it is a hammer. If we are to licitly use animals in medical research, our actions must be consistent with the grace-infused fruits of the Holy Spirit. Medical research on animals, therefore, must make a real effort to allow these animals to be the kinds of creatures God created them to be. And the more social and sophisticated the animal, the greater this demand is. Though I'm very happy to be corrected by those actually doing practical work in this field, like, say, our moderator, because dementia comes about and manifests itself in living bodies, I very much doubt that neural organoids will do what they need to do in terms of reflecting predictable results of most therapeutic interventions. Researchers will most likely need to move to neural chimeras to get better results and better data. And obviously there will be a temptation to use animals that are more social and more sophisticated and have brains that are more like ours to get the best results possible. I propose that because the creation of neural chimeras fundamentally alters the kinds of creatures involved, violently and dramatically removing them from the telos with which God created them, they are not examples of licit medical research and not at all consistent with the clear Catholic teaching that we owe animals kindness. Because of this, we must find other ways to research these terrible diseases. And again, to be ready with an all-hands-on-deck approach to care for people with dementia in the event that the next decade or two does not produce the therapies we are hoping and praying for. Thank you.
Thanks. Thank you to my good friends Charlie and Kristen for such rich and thoughtful interventions. I was sheepish to accept the invitation, Kristen's invitation to participate in this panel discussion. No, the joke comes later. I'll finish, after we talk about the dark stuff, we'll turn to the joke to bring us back to a lighter direction. But because this is the Nicholas Center's conference, it's always weird to like, oh, I'm gonna put myself on a panel. That's not what I'm doing. And I was, in any event, so how to think about this? I think a useful framing document has already been mentioned by Kristen. The National Academy of Sciences had in 2021, convened a working group. The actual working group itself was the Committee on Ethical, Legal, and Regulatory Issues Associated with Neural Chimeras and Organoids, the Committee on Science, Technology, and Law, Policy and Global Affairs, all associated, of course, with the National Academy of Sciences, Engineering, and Medicine, the most august and esteemed body of its kind on the medical science and other science landscape. And it's a useful document in several respects, in the sense, first of all, it's useful as it draws out a kind of a rough sketch of the regulatory apparatus and the goods that those mechanisms aim to serve in this space. They conclude, of course, that there's not sufficient mechanisms of oversight for this area. It's so novel. I mean, there's some overlapping concerns with other areas of research involving informed consent and animal welfare considerations that go into research involving other animals, which may or may not be adequate, but they flag those issues. On the question of it's a useful point of departure because they flag 7 ethical issues, but then in some ways, and this is not probably, it would be too much to ask for them to resolve all the puzzles that these ethical concerns surface, but what I'm going to do is briefly name the ethical concerns that they talk about, and then talk about the puzzles that they gesture towards, definitional puzzles, deeper normative puzzles, and then make a few suggestions for how we might think about this going forward. On page 97 of their report, they flag, in the summary of chapter 3 on ethics, they say, There are 7 reasons why, and I'm just going to paraphrase. First are the ethical reasons for working to relieve serious human suffering caused by illnesses and dread diseases and injuries that have already been discussed by the panelists. The imperative to care for one another in that way, and the utility of all of the different kinds of entities we're talking about here as models or even therapeutic interventions to care for people in those situations. Secondly, they say, there may be concerns about human beings encroaching on divine roles or overreaching appropriate human activities. Now, there's a way to say that, I think, that's rich and deep, the way Dr. Collier did. I was troubled a little bit by the way it was framed in this report, because it seems to, and it's hard to tell when the report is taking a normative posture and saying what things should be versus simply describing the opinions of folks in the public square. And in this instance, it felt to me like that way of describing in some ways diminished the force of the objection, made it seem like it was peculiar to people with particular religious points of view, and in confronted with the problem of pluralism and a pluralistic system, we of course respect those folks, but we don't inhibit progress in the name of other people's theology. I think there's a lot more. I think there's a deeper way to talk about the kinds of issues that have been flagged by our panelists, and so I would quibble a little bit with the way they described that. Third, there may be ethical issues involved in carrying out such research using cells derived from biological materials of persons who didn't know they were being used. That's a garden variety informed consent problem. That is not necessarily distinctive to this particular area. Fourth, there are various concerns related to the distinctions between human beings and other animals. Well, that's certainly true. Fifth, there are concerns about animal welfare and animal rights. Sixth, there are concerns about consciousness and enhanced capacities in research animals and neural organoids. And finally, there are concerns about the use of non-human primates in such research, which again overlaps with other areas of concern in medical research. So they're worried about, and in particular, if you dig into the text, they focus on the problem of consciousness. They talk about consciousness as something that matters a lot to doing an ethical analysis of the moral status or the moral standing of engaging research involving neural organoids, cerebral organoids. They talk about pain. And then they talk about species boundaries. Species boundaries in terms of not just the mixing of animal and human, which raises its own kinds of questions, but also species boundary issues implicated by the possibility of enhanced capacities, and these relate to the brain and the nervous system, or reproductive capacities. Right? So brains and gonads loom very large in this report, meaning in the sense that imagining an animal who has, by virtue of the fact that the animal has a brain that the structure and function of which is human or is composed entirely of human neurons and glia, is that a problematic state of affairs to have an animal like that? And here's the joke. How do you know if that happens with a mouse? You show up and he's fashioned a diaper to cover his nakedness when you arrive the next day. It's not a great joke, but I didn't want to say there are no jokes, but in any event. But imagine the scenario in which a non-human primate had their germline populated with human reproductive cells. Meaning, imagine a non-human primate that could produce human, let's say, sperm or eggs, human gametes, and of course functional human gametes, which could lead to the reproduction of subsequent offspring. This is an ethical issue that's flagged by the National Academy. Now, again, it's asking too much to ask them to drill down into the depths and complexity of all these issues, but that's really a job that we have to continue if we're going to be serious about thinking through these issues, not just as ethical matters, but in terms of public bioethics, governing ourselves, medicine, science, and biotechnology in the name of ethical goods. So the first thing that is in some ways unsatisfying about the report, and it probably was unavoidable, is that there are definitional puzzles that emerge. First of all, the issue of consciousness. They don't define consciousness in this report, and of course it's contested, and there are many different theories of consciousness, and then there are sort of related or adjacent concepts like sentience or self-awareness, and then even further adjacent concepts like empathy and altruism and imitative learning and joint attention and these kinds of concepts. It seems to me that if you can't have a functional definition of consciousness, you can't identify the neural architecture necessary for it, and you certainly have serious difficulties in developing tests to identify it, right? And so I think this is a significant problem in the context of neural organoids. I do, and maybe it's because I don't, when I'll dig into the science, my concerns will be allayed. But I do have concerns about brains, or I don't want to be inflammatory. Let's use the language, neural organoids that have patterns of activation and function in ways that mimic cerebral tissue in vivo. And there's one example of an experiment that was, and of course there's a deep question, can you have consciousness without experience? And this was Charlie's important point. Well, apparently, in August of 2021, scientists developed cerebral organoids that grew eye-like structures. They had optic cups, and there was some evidence of visual processing in the organoid itself. Now that, in some ways, points us in a worrisome direction in terms of thinking about what exactly consciousness requires by way of experience. Is visual perception part of the issue? And then I've heard just before we got started today about an organoid that was connected to a nervous system that was also connected to some musculature, and there was movement that was originating in the organoid that went through the nervous system and moved the muscles. So that to me points to a very complex question. The issue of pain, they talk about. There are definitional questions involving pain. Pain is a contested concept too. Some argue that pain is merely a physiological reality. Others argue that it's necessarily a psychic reality, and you have to have certain kind of cortical structures in place in order to experience pain. Some capacity for reflection or awareness. And this actually plays out in dramatic fashion in the debate over fetal pain in the context of abortion. At what stage of biological development of the unborn child is the capacity for pain real? It's a definitional problem. And of course, there's a definitional problem about humanness itself. What exactly is a human being? And by extension, what are the boundaries of the moral and legal community? There are questions about the definitions of organisms and issues of parts and wholes, right? I think most of us would have less concerns about a kidney in vitro setting than that was functioning in some way as kidneys do in the organism versus a neural model, three-dimensional neural model, the likes of which we're discussing now. So the definitional problems, but even more deeply than that, the problem that emerges from reading this report is, why do we care about consciousness and pain and species boundaries? Why are these the questions that we're asking in the first instance? And it won't surprise you, for those of you who are familiar with sort of how I see the world and how I think, that I think any kind of set of normative questions that are meant to guide us towards an ethical conclusion, and certainly by extension a legal and policy conclusion, are rooted unavoidably in anthropological premises. What does it mean? What is the idea behind human identity and human flourishing? Not the boundaries question, but the question of everyone in this room. Who are we, and what is our flourishing, and what do we owe to one another? There are presuppositions that are animating us and leading us to the questions about consciousness and pain and species boundaries. And my argument that I've made elsewhere, and it very much tracks with what Alistair was talking about in his remark, is that any anthropological account that doesn't take seriously our embodiment and is strictly cognitive and strictly associates people as a kind of atomized individual will whose flourishing is to pursue his or her own internally, endogenously generated dreams and plans, is going to miss huge. I mean, it's true in part, but it misses a major aspect of our lived reality, which is our embodiment. I agree with Alistair very much that we are bodies. We don't merely have bodies, and the fact that and the insight that we are bodies situates us in a certain kind of relationship to each other. As bodies, we are subject to illness and dying and injury and senescence, and stands us in relationship with each other, in relationships of mutual dependence. Our vulnerability stands us into a kind of relationship with one another, mutual dependence, and which then points the way to human flourishing and what it requires, namely networks of what Alistair calls networks of uncalculated giving and graceful receiving. Not freedom of the unencumbered will, but rather freedom to care for one another rightly. And so I worry about any normative framework that starts asking questions about and prioritizes cognition in the way that this framework does, because it does actually seem to track a different kind of anthropology, an anthropology that stands that conceives of human flourishing as an atomized will trying to impose its preferences and pursue its dreams over and above, perhaps in collaboration, but also in conflict, in a world of strife. And it seems to me that that kind of anthropological set of premises leads to attitudes that are very extractive and exploitative to not just other people, but to the natural world around us and to animals too. That if we see the world around us as inchoate matter, including our human relationships, through the lens of pursuing our own authenticity, our own unique dreams, then it seems to me that we're going to lack the resources to care for one another the right way. The report also suggests that we try to identify the criteria necessary for moral status. I think of that as a failed project. I think of the personhood project as a failed project because, first of all, it began as a kind of exercise of dividing the world up into who counts and who doesn't for the purpose of excluding a segment of the human population that we wanted to have an extractive relationship towards. That we want, and that's not limited to people either. It could be limited to, it could also include animals and the natural world. And I think one insight that I think we should focus on here when we're talking about chimeras especially is we shouldn't confuse the criteria for membership in a species. That is to say, it might be okay to say human beings, one of the criteria for membership in the species has to do with something, the potential capacity for rational thought or a rational nature with the grounding of the moral value of that individual itself, okay? It's a different thing to say that rational nature is what makes you a part of a particular classification scheme. It's different from saying having a rational nature is what makes you valuable and inviolable. Those are different things, it seems to me, and we should keep them separate as we think about these chimeras. And I'll conclude by saying, I don't think the problem of membership or the problem of boundaries, even if we come to the conclusion these aren't organisms, these aren't human beings, these chimeras aren't human in some important way, that doesn't settle the problem of what our obligations are. And I think reflecting on the concept of vulnerability and what it means and how vulnerability plays out in our own interpersonal relationships has insights for us that we need to think about when we're dealing with the natural world, we're dealing with animals, and even the environment itself. The idea that vulnerability is a kind of summons for care. It's not an opportunity to exploit and extract. And it seems to me that the concept of stewardship, rather than ownership and rational mastery, is the right principle to think about when we're talking about these questions. And we have to ask the question not about animal rights or does this animal deserve that, but rather what does it do to us? What does it do to our soul when we adopt a purely extractive attitude and posture, not just towards other people, but towards animals and the natural world? And so I think there are no answers in my very brief remarks, but I think reflecting on human vulnerability, what it means and how it stands us in relation to others, and including non-human animals, gives us some pathway forward in thinking through these questions.
At this time, I'd like to take moderator's privilege to say a couple remarks of my own and pick up just on some threads that you guys all spun. A few things that I'd like to pick up on. One, both Dr. Collier and Dr. Kamosi, you pointed to this reality that our results from these experiments are only as good as the models that we have. And you asked the question of whether these models will be sufficient to learn what we think we want to learn about these various neurological diseases, what it is that we'll actually get out of these models. Some things that cerebral organoids may be helpful for learning about are things like learning about the ways that cells migrate across the developmental field as they start to separate out and differentiate into their various cell types. How is it that they're interacting with one another? How is it that they're migrating in these little organoids, which are like 3 or 4 millimeters in diameter because they don't have vasculature yet? Scientists are already starting to notice that certain cell types can send projections way out. In this example that Carter Sneed pointed to about the cerebral organoid connected to a spinal cord organoid connected to a muscular organoid that made the muscle twitch because of the spontaneous activity in the cerebral organoid. the scientist, and this was done at Stanford, my home institution, Dr. Pasca noticed that certain cell types were able to send out projections to the other tissue types kind of innately. So organoids are shedding a lot of light on the intrinsic ability of each cell type involved in the model, at least certain intrinsic abilities, to see what can they actually accomplish by themselves outside of the organismic context. And it is quite impressive that these stem cells retain the ability to self-organize in these very complex structures that do mimic a lot of the structures that we find in vivo. So that's one thing that organoids will be helpful for. As Dr. Collier pointed out, they are living models, which is better than our autopsies and other dead tissue models. But then chimeras add this layer of being an organismic model. So not only how are these cells innately acting according to their own potencies, but how are they also interacting with the rest of the organismic structure, whether that's anatomical structures, other cell types that aren't necessarily derived from the same progenitor cells. And so the chimera provides more of this organismic context. But again, the organismic context is quite idiosyncratic for each species. And so then we'll butt up against experimental problems with, well, how does this actually work in the human organism? And so I see as much as organoids and chimeras are being celebrated right now in the scientific community, one thing I do worry about is that there may be a pressure, again, to come back to the question of using human embryos and being able to maybe mass produce them or to at least observe their development up to certain points. And of course, scientists are always trying to push that boundary past 14 days, past the last barrier we never thought we would get to. So that's one concern I have. Second, I do want to make an optimistic comment to say that there are non-religious scientists who are also very concerned about some of these ethical questions brought up. I know I'm kind of backtracking back to the ontological questions that we started with at the beginning, but there are scientists like Hiromitsu Nakauchi at Stanford from Japan who, because he's worried about donor cell contribution to a chimeric animal's brain or gonads, came up with the bright idea of first genetically modifying the donor cells before inserting them into the chimeric organism to knock out a gene that's critical for their incorporation in the brain. So these cells will then go on to contribute in some small proportion to every tissue in the animal's body, but is prevented from incorporating into the brain because it kind of missed that stop on the train. It didn't have that gene to go down that pathway. And I haven't heard him say that there's an analogous gene for gonadal incorporation, but perhaps there is something like that. Dr. Collier mentioned blastocyst complementation, which is kind of the inverse of what I just described, where the animal embryo host has been genetically modified before the chimerization event, and the host species has been knocked out for some gene that's critical for developing a particular tissue or organ type. Many of the studies have been done with the pancreas, because PDX1 is a gene in mice that when you knock it out, it just completely eliminates the development of the pancreas. So if you do that in a mouse embryo and then inject donor pluripotent cells, whether from a rat or another species like a human, then when it comes time for the organism to develop its pancreas, it's not able to do so with its own native cells, and the donor chimeric cells then occupy that developmental niche. But Professor Nakauchi also pointed out to me that not every organ has a single gene that controls its development, like with the pancreas. And so it's been very difficult to create things like organisms with a fully chimeric heart because knocking out key cardiac developmental genes so influences the development of other systems as well, that the organism isn't viable or your model is totally ruined for other reasons. So this possibility that we sometimes talk about and that a lot of the mainstream ethicists like to speculate about in the literature, of having an animal with a completely human brain, a human-derived brain, may not even be a scientific possibility if there's no kind of single gene or few discrete genes that only affect neural development and not the rest of the organism. The last point that I wanted to make is about this question of natural kinds. As it has been pointed out, this National Academies of the Sciences, Engineering, and Medicine report focused a lot on threshold capacities for the determination of moral significance. And Professor Sneed kind of alluded to a way around this, that it's not necessarily what we do that makes us matter morally, but what we do is a clue to the deeper reality of what we are, who we are as human beings. And so one question that I have been engaging with in my research on these topics is whether just the creation of chimeras totally disrupts a traditional understanding of natural kinds. Does it introduce some kind of major philosophical wrench and bring gradualism straight into the forefront of our minds? That is all of biological reality just one continuous spectrum where we can't distinguish morally relevant kinds anymore? But I would like to put up a picture that I find the most striking illustration of this. And I'm taking a lot of moderator's privilege. Can you see this? So this picture really changed the way that I think about the science of chimeras. On the left side, you'll see a mouse. On the far right side, you'll see a rat. And then the two organisms in between are both rat-mouse chimeras. The one, second in from the left started as a mouse embryo and rat cells were injected into it. The one just in from the right started as a rat embryo with mouse cells injected into it. And the thing that really struck me is that this is not a continuum of biological being. The chimeras basically obey the basic body plan of the host organism. So I've called this the graft-host paradigm of body plan dominance. And this, combined with many of the interspecies barriers that Dr. Collier alluded to, like cells maturing at different rates in different species, cells using different kinds of signals to communicate with one another, differences in anatomy, physical pressures, and electrical stimulation in each organism, means that it's going to be really, really difficult for us to actually find stable biological patterns that break from what the evolutionary loom has already woven for us, just because the organism is so complex. And so Professor Nakauchi told me, kind of off the record, I don't know if he's been able to publish this research yet, but in his creation of chimeras, organisms that started to exceed a certain percentage of donor contribution, they would just die in utero because the biology couldn't handle having like 50%, 50%. They really are tending towards these different stable configurations. And so I offer this picture also as, for me, an optimistic note that the science is pointing to in terms of the lasting validity of our traditional notions of natural kinds. So thank you for permitting me that. We still have several minutes for questions for our panel. So if you have a question, okay. I want to give Megan Levis the first question. She gave a great talk yesterday on organoids.
All right, so I have two questions queued up, and I'm intrigued to hear which one you would prefer. So I have one on the soul and one on AI.
Soul. We want the soul.
So, okay, great. Awesome. I guess if you wanted the AI one, you could have come to my talk. This is something, so I've been thinking about organoids for a number of years now, starting with my PhD work and now kind of moving into my postdoc work. And one of the questions that I've kind of run into, especially as we're talking about neural organoids, is sort of like related to the mind, is like this question about the soul and like the rationality of the soul. Question that I've typed out here is that organoids seem to be something that's like not quite human, even if they're made out of human cells. But when we're considering questions about consciousness and reality, the soul seems like really important to this kind of discussion. Do you guys have like thoughts on that, or kind of like an inkling of like an indication for kind of how we should be thinking about that? Because this seems like very much related to the questions of kind of human dignity.
So as I mentioned in my remarks, there are lots of people. In fact, there might be some people in this room who might want to chime in who are trained in philosophy of mind and other related disciplines to offer their thoughts on this. But you know, it's interesting, the traditions going all the way back to the marshalling of Aristotle's notion of the soul as the form of the body, I think looms large here, in that it really correlates with so much of what Alvin Noe and other thinkers in philosophy of mind are thinking of now as consciousness as arising from the organism holistically considered in relation to their environment. So to the extent that we think of the soul as connected to consciousness this way, I just think there's just a almost embarrassing amount of riches for work to be done from the tradition, a hylomorphic tradition on this, to try to fuse these two kind of conversations and think about this together, which is why, though I'm happy to change my mind based on the things, you know, Carter's given me some things to think about here, obviously, which is why I'm just at least still not at all worried that we're talking about consciousness here, because it's a product of the holistic organism in toto.
Yeah, just very briefly, I think the concept of organism is really important to this discussion. And the question is posit a cerebral organoid that had the capacity for sensory inputs, and you can measure patterns of activation in the brain that seem to correlate with visual perception and processing, just for example, or some other mechanism of receiving sensory inputs. It seems to me, again, on one foot, like it's not a human organism in the same way that an embryo or a fetus is a human organism. But if we were to encounter some alien life form that was in the form of a cerebral organoid that had the capacity for sensory inputs and patterns of activation that looked like something was happening, I would be concerned about an exploitative and extractive attitude towards that kind of a being. I don't know what I'm supposed to do with that being, but I think, and again, you borrow from mostly European environmental regulation concepts. The idea of a precautionary principle seems important here, because in the NASEM report, there's a lot of talk of, well, we can't do this now, and this probably won't be possible. But that to me, or even worse, if it happens, we'll be sure to kill the thing before it gets too big. That's not. I don't find that ethically comforting, right? I just think that we have to think about these things and posit, a theory of what they are that entitles them, that prevents us from that sort of little off ramp to make life easier on ourselves.
Yeah, thank you.
You didn't get your question for Professor McIntyre, so here you go.
Thank you all very much for very interesting comments. I'm Adam Myers, Mount Mercy University, Cedar Rapids, Iowa. One of the elements of most of your talks seem to be concerned about certain kinds of things that we're able to do now that we feel we shouldn't do, in experimentation and other kinds of things. but I have a hard time envisioning what is it like, sort of culturally, socially together, to not do something that we think would have very good consequences, right, in terms of medical outcomes, curing, therapy, so on and so forth. What would it be like as a culture to not do something that we're capable of doing? How can we envision that or imagine that? I mean, part of the justification for me of the sense of this question is that most of the stories we tell about technology and ability are about like how great it would be if we could do this, right? A lot of science fiction seems to be sort of geared towards envisioning a world where we've done these kinds of things. Or they're dystopian science fictions where it's like envision doing this and see how horrible it's going to be when you drive nature out with a pitchfork. It's going to come back and terrorize you. And I'm just wondering if there's more positive visions of what it's like to know what it would be like to actually just withhold ourselves from doing these kinds of things.
So I'm going to hand this to Dr. Collier in a second, but I just want to say a very brief thing. We already restrain ourselves in dramatic ways based on ethical principles. It's just that we have a shared understanding of harm. So informed consent is a massive restriction on what we could do scientifically. If the integrity of human bodies is a massive constraint on what we could do. If we didn't observe the ethical norm of informed consent or even basic principles of self-determination and autonomy and human subjects of research, we could solve a lot of problems a lot more quickly than we otherwise could. So we do, in fact, restrain ourselves. The question is, how do we deal with these areas in which there's deep disagreement?
No, I don't have anything else to add to that. Thank you, Carter, for that response. I actually have my own question. I'm just going to just pull panelist privilege, I guess, actually, because we haven't talked about this. So actually, Charlie, I was so encouraged to hear that the committee had, people like yourselves and from a theological perspective to give their voice into this important subject. I guess, how were your comments sort of regarded by the committee? I saw when I was reading the report, like several footnotes that were added by comments that you made. Do you think that your comments were held to the same sort of standard or just to the same sort of legitimacy that they gave to other folks on the committee? Or how were you received as the theologian on this group?
Well, it was a pluralistic venture, so they had other theologians and religious figures and philosophers and others that they asked. But I went in with that question myself. I think it's true that Jerry McKinney of this Augusta institution was going to be asked to do this, and he kind of sloughed it off on me, which is how I got asked to do it. But he's a very prominent, very wonderful Christian moral theologian, and so they wanted this kind of input. They seeked it out, and I was treated respectfully. I was, sometimes they kind of, but it helped that there was this part where they were seeking out religious voices as part of their explicit, testing ground here. And I received very positive feedback. I got some pushback. One person asked me why I wasn't against mules for reasons you might imagine. Well, they just assumed that I wasn't, we didn't really get into the missionist thought about this very deeply. But that's neither here nor there. But I mean, this is a long answer to a short question. I mean, I was very impressed, frankly, that they were interested.
Kristen, when we had the President's Council on I was the General Counsel of the President Bush's Council on Bioethics under Dr. Leon Cass, who was later succeeded by Ed Pellegrino. And it was a very interesting, different approach to this question. Whereas NBAC, which was President Clinton's Bioethics Advisory Committee, they followed this model of the National Academy. They would bring in like a panel of different theologians from different traditions. Leon, his view was that it's better to have, and this was actually how bioethics emerged in the first instance, theologians and moral philosophers and politicians and lawyers and bench scientists and physicians and social scientists all at the same table deliberating as the deliberative body itself, rather than serving as a kind of expert witness to professional bioethicists. And I'm not objective because I worked for that body, and I think Leon Cass is a wonderful person and a brilliant man. But I think in some ways, I prefer the model in which there's integration of theological minds and voices as opposed to bracketing them out as a sort of, well, here's a panel of of different religious leaders or religious thinkers, and we're just going to hear from them, and so that we can describe the landscape of different opinions, which is what I think this is the model.
Now I have to do what every moderator hates to do, which is to cut short a very interesting, stimulating, even if weird conversation. Although I think it's probably more important that we go worship our Lord in the Holy Mass. So thank you all for joining us.
Dylan,
Please-
can I take the director's privilege real quick? I just wanted to say something very briefly. Moral philosophy, theology, other sources of knowledge and wisdom are really important, but I think we can also get a lot of wisdom from literature and art in this domain, and there's a kind of sense in which we can understand the human reality of these questions in a deeper way through artistic presentation. So to that, I just want to recommend a book from 1953 called You Shall Know Them by Vercours, which is a very French book in the sense that it's about a man who impregnates a non-human primate. There's an offspring who he then murders and throws himself on the mercy of the court and demands to be prosecuted for homicide. And the entire book is about that trial. So I just want to say we can't neglect artistic sources of wisdom when we're thinking about these very complicated questions.
Just when you thought it couldn't get any weirder. So please join me in thanking our esteemed panelists.